Hello, I'm Mr. Baker, and this is Unit 2, Biochemistry, or the Chemistry of Life, and this is Sections 2.3 and 2.4. Hi, I'm Mr. Baker, and this is Unit 2, Chemistry of Life, or Biochemistry, and this is Sections 2.3 and 2.4. The direction of osmosis. We introduced osmosis at the end of 2.2, and the big question about osmosis is where do the water molecules move? And that comes down to which side of the membrane has more solute or more stuff. We're going to break this down simple for you. I like to teach this concept, and it is psi, P-S-I. It is that little weird-looking trident symbol. It's a Greek letter, and they've adopted the Greek letter, and we're going to refer to it as psi from here on out, and it is the new osmosis because, quite honestly, I can never remember those three tonics. However, psi, I never screw up. It is pronounced psi, and it's a Greek letter. It is known as water potential. Water potential is the tendency of water molecules to move to a particular area. There are three rules of psi that you have to commit to memory. And if you can do this, how water moves around is quite simple. The three rules are: the psi of pure water is 0. Pure water meaning there is nothing in it. I'm not talking tap water, I'm talking pure water. The more stuff you have in a solution, the psi is more negative. Water will always move to where the psi is more negative. So therefore, water always moves to where there is more stuff. Psi is involved in water transport. Water will always move to where the psi is more negative, and osmosis is this passive transport of water across a membrane. The psi of pure water is 0. So what is tap water? The psi of tap water is slightly negative because tap water, even though you don't think so, has a small amount of stuff in it. That's why it has taste. Those are some dissolved minerals. It's not pure water. Pure water is distilled, and even distilled isn't perfectly pure water. Salt water is much more negative because salt water has a whole bunch of stuff in it, the stuff being salt in this case. So, can I drink salt water? If you're lost at sea, there's a whole bunch of water. How about I drink some of that? Well, if you're lost at sea, the psi of the salt water is more negative than the psi of you. So if you drink salt water, you're going to pull water out of your cells and into your stomach. And in fact, the Dead Sea is so salty that it will pull so much out, it can kill you very quickly. If you continue to drink seawater, it will also kill you. So this osmosis and psi stuff can be very important. Can bacteria drink salt water? The question came up. So we got some bacteria that got lost at sea. Can they drink seawater? Well, it turns out that the psi of bacteria is about negative 0.3. The psi of seawater is negative 25. Which one is more negative? Correct. Seawater at negative 25. So the water is going to move out of the bacteria and into the seawater, meaning the bacteria is going to have a hard time living without any water. Well, what does this mean for me? Well, there's all kinds of psi in your pantry and in your refrigerator. What do these all have in common? Pickles, which were invented about 2030 BC in India, and jerky, which was invented about 1500 AD by the First Nations of South America. And what they've done is they have used a whole lot of salt to keep bacteria from making their food go rotten or spoil. I like cucumbers, but you can't leave a cucumber in your refrigerator for very long or out on the counter because it goes bad. But I can put it in pickle brine, which is a whole lot of salt, and it keeps the bacteria, and that cucumber can survive for a much longer period of time in my fridge with a whole lot of salt around it than if I leave it by itself. Same deal with meat and jerky. Same deal with sugar, because humans invented that, and it's attributed to the Romans at about 100 AD, jams, jellies, and marmalades. So instead of using salt, they pack a whole lot of sugar, and you can do this for grapes. You know, I'd like to have grapes, but you can't keep them around very long. But if I smash them and pack them in a whole bunch of sugar, then the bacteria can't live there. For this reason, small increases in the salt content or salinity of lakes can have disastrous consequences for organisms living there, from fish to bacteria. Here's a picture. On the left, it's a plant that's in salty soil, and the picture on the right that's in normal soil. And that's, if the plant doesn't get enough water, that's a bad deal for the plant. Made a table for you. We've given quizzes over this in the past, and perhaps will again this year. Here's a nice picture showing the various tonics and what happens, and there are simple experiments you can do at home. Take celery, put celery in salt water, it gets all floppy. Put celery back into regular water, and it gets all stiff again, because that's the amount of water that goes in and out. If I put a cell into, and then we're looking at the left-hand picture, into pure water, the cell will expand, and boom. If I put it into salt water, middle picture, the cell will shrink, and if I put it into the same type of solution with the equal amount of psi, the cell will stay its normal size. Acids and bases. Acids is a substance that can increase the hydrogen ion, or H+, of a solution. They tend to taste sour. Lemon juice, orange juice are examples of acids. Also acetic acid, your vinegar. A base is a subset that increases the hydroxide ion, or OH-. These taste bitter, they feel slippery, and it's also called alkaline. NaOH is also referred to as lye. Soaps are basic. Bleach is basic. A few foods are basic. Most of your foods you're actually going to eat are acidic. Here's a pH scale going from 0 to 14. Due to math, it's 0 to 14. The closer you are to 0, wow, are you acid. The closer you are to 14, wow, you're a base. And the closer you are to 7, you are almost neutral. pH, it's a measure of the concentration of H+ in a solution. P for percentage, H for hydrogen ions, ergo pH. Like I said, it's from 0 to 14. Anything from 0 to 6.99999 is acidic, meaning that you have more H+. If you are exactly at 7, you are neutral. You have equal amounts of H+ and OH-. And if you're 7.1 to 14, you're basic, meaning you have more OH-. And here's a table, or a little picture. It's got some examples of acids and bases. In order to survive, humans must maintain a pH of 7.35 to 7.45. That is slightly basic. And this brings in the concept of homeostasis. Homeostasis is maintaining a certain level. If your pH goes way lower than 7.35, in fact, if you just drop down to 6.8, that's enough to cause enough things to go wrong that it will kill you. If your pH goes up to 7.8, that's also enough. So you want to keep your pH, your blood pH, around 7.35 to 7.45. That's homeostasis. Chemistry of acid base and antacids. If people have serious acids in their stomach, which is a pH of 2.5, sometimes that can cause problems, as the mucus layer that protects it can be eroded away. And what you're going to do is you're going to take an antacid, which is a base, and you're going to try to counteract that, which seems simple. However, you can overdo antacids, and evidence was showing when I was your age, older males got ulcers, and it was thought because of stress. Well, it turns out that ulcers were caused by bacteria that were breaking down the mucus layer. So then we just put people on antibiotics, and antacids aren't nearly all over the place like they were when I was younger. There's a picture of the stomach. That takes us to quiz 2.3 over water potential, the various tonics again, the psi chart, and acid, base, and pHs. Section 2.4 takes us to organic compounds, which are things that contain carbon, and they are the 4 major macromolecules, including carbohydrates, proteins, lipids, and nucleic acids. Here are your biomolecule building blocks. Nucleic acids have nucleotides. Carbohydrates are monosaccharides that get built up. Lipid is glycerol and 3 fatty acids. And proteins are chains of amino acids. These are complex compounds in food, such as proteins that are broken down or built. How do they break down and build? You need to know the difference between a monomer, mono means one, and a polymer, poly means more than one. Dimer means two, that's two monomers. Poly, three or more monomers joined together, and it's like, we came up with this a couple years ago, a train. A train is a long chain of cars. The train is a polymer, and the individual cars that you attach to them are the monomers. You can have long chain, or a long train in this case, or you can have a short train. The first major molecule are carbohydrates. They get their name because they're composed of carbon, hydrogen, and oxygen, hence carbo and the word hydrate. Carbon, hydrate refers to water, carbon, hydrogen, and oxygen, which are in water. The monomer here is called a monosaccharide, which I expect you to know. Mono means one. Saccharide is a fancy name for sugar. They are the essential sources of energy for all living things. So if you think carbohydrate, think sugars and starches. They are stored in the chemical bonds. They like to dissolve in water. They are hydrophilic, which means they love water. That's why you can dissolve sugar into water and make sugar water or Kool-Aid. Most carbohydrates are ultimately converted into glucose to make ATP, and ATP is the energy molecule that your cells use. More about that in later chapters. If you want to store them, you can store them in larger complex molecules like starch, which is 100 or more glucose molecules joined together. That is, starch is a polymer, glucose is a monomer. Here are some other. Fructose is a simple, and the complex in I guess it's oatmeal. Looks like oatmeal. It's a long chain of starch, so that's a polymer. Not all carbohydrates are digestible. Chitin, which is found here in lobsters, and in also cellulose, which is wood. So there's a picture of a lobster, which is chitin, and wood has cellulose. Although it's good to have extra fiber in your diet to keep your digestive system working, but more about that later. So don't forget, a monomer, glucose is a monomer, and several polymers put things together. So starch is a polymer, cellulose is a polymer, glycogen is a polymer. It's just how you build your train. That takes us to lipids. The monomer is something called a fatty acid. These guys are nonpolar. They do not dissolve in water, so they are hydrophobic. And hydrophobic means scared of water or water-fearing. And these guys are greasy to the touch. There are three main types: fats, sterols, and hormones that we expect you to know. There's a picture of a penguin because it looks cool. Fats store more energy than carbohydrates, and they involve triglycerides, which are fats in the most foods that we eat. They're composed of one polar head and three nonpolar fatty acids tails. So look at the picture here. In the blue is the glycerol head, 3 carbons, joined to 3 long chains things that are called fatty acids because they're acidic and they are fats. For us, what do you need to know? That there are 2 main types of fat, saturated and unsaturated. Saturated are all single bonds and they're solid. There's a whole bunch of S's there. That's your mnemonic device, Unsaturated fats have some double bonds. These guys are liquid at room temperature, like olive oil and vegetable oil. Again, here's another picture of your saturated fats, which tend to be solid at room temperature, and your unsaturated fats, which they should be a picture of oil there, but they don't have one. All of these are composed of an even smaller piece called a phospholipid, which can consist of a head and a tail, the phosphate head and a fatty acid tail. That head likes water, hydrophilic. The tail doesn't like water, hydrophobic. And this is how we form something called a phospholipid. Phospholipids then can form around and squeeze out water. They are the basis of membranes. See if you can remember that. Another one is waxes. These are water-resistant coating on many plants and animals. Yes, in your ears, that's a wax. Sterols play an important role in growth and development. Cholesterol, component of most cell membranes. Too much cholesterol can block your arteries, so you want to make sure you're not eating too much of that. Steroid hormones can also help growth and development, like estrogen and testosterone. There's another picture of your sterols. Sterols all have, do you see the middle of that picture where it's almost stop sign, almost stop sign, almost stop sign, and home plate? That is a general structure for all sterols, and they just have other things around the outside. So look at underneath that picture, estrogen and testosterone. See how they are slightly different based on one's got 4 things, testosterone coming off of the basic, and estrogen has 3 things. That takes us to quiz 2.4, which are carbohydrates, which are the sugars, starches, and lipids. And that's what we have for now. Thank you. Study hard.